Many thanks to all for coming, and welcome to all our second seminar this year, which has been already one week after the first one, to present Ricardo Amils amongst astrobiologists seems to be a little bit redundant because very probably we have all met him at one of our meetings. At the last one I met him was actually our Beacon conference. He has got his licentiate in the University of Barcelona in 1969 and then did a thesis on the biochemical department on the Faculty of Medicine of the University of Buenos Aires in Argentina before becoming a Doctor of Science at the Autonomous University in Barcelona. Today he's a full professor at the Autonomous University of Madrid and mostly working at the Centro de Astrobiología in Spain. And most of you probably will know Ricardo from his work on extreme environments like Rio Tinto, the Dalol Plain in Ethiopia, and the Salar de Uyuni. So this was my contribution today in the afternoon. Please enjoy the talk. And now I would like to hand over the word to Ricardo. Welcome to our second seminar.
Okay, thanks, Wolf. And I'm sorry that this will be an anonymous presenter because I just got a new computer. Yesterday we checked it and it was working all right, but today the camera doesn't want to work. But I think what is more important is that you see the kind of work I have to share with you. So I go ahead. I will try to entertain you for this afternoon talking about the dark biosphere. The talk. I mean, it is well known that Darwin is not only famous for his work on evolution, but he always made many other observations while he was in the Beagle doing his trip around the world. And he explained in his travel books that arriving to Argentina, in the interior of Argentina, to Salt Lake, he took some samples. And then later on, when he was in the boat, he inspected them on the microscope and he saw that they were was life in a very extreme allophilic system. Probably he was looking at Artemia salina. And he made the following prediction, which I think underlines the geniality of Darwin. He said, if there is life in an environment like this, it should be life associated to the volcanic activity in the summits of the highest mountains, under the polar ice, under the deep sea, and also in the deep subsurface. I mean, in the under our feet. From probably what you saw is seeing all these scenarios that most of the predictions of Darwin has been done, and nowadays this is an interesting subject of microbiology, what is called extremophiles. I mean, the extremophilic environments. But it took a long time to do the kind of work that is suggested in the deep subsurface, and that's the question: why it took so long? And mostly it was because the scientific community was very skeptic about the possibility that there was life on the hard rocks in the continental subsurface, and also because some preliminary data that were obtained in the beginning of the last century in doing oil drilling, I mean, it was considered that that was due to surface contamination. So for many years, I mean, this kind of work was not considered until 20 years ago. Was published the first paper describing life on the hard rock continental subsurface. And why we became interested in the dark biosphere? I mean, well, probably some of you know that we've been involved in studying the geomicrobiology of Rio Tinto for more than 30 years. And Rio Tinto is an acidic river, acidic pH 2.3, almost 100 kilometers long, with high concentration of ferric iron, which is in solution due to the acidity of the system. Rio Tinto rises in the heart of the Iberian Pyrite Belt, which is the highest concentration of metal sulfates on Earth. And I will just try to make very short history of our past research, the, checking some of the advances in the microbiology of the system. In the beginning, we were only able to use isolation, microbes isolated from the river to study their characteristics, but we have to accept that the eruption of molecular biology techniques has been an authentic revolution in the study of extreme environments and obviously in acidic environments too. And here you can see in the bacterial tree of life, stated in red, the phyla in which we have identified or isolate some microorganisms of the system. Also, we tested the many different techniques. One of them, you will hear more about them later on. It's a fluorescent in situ hybridization, which I like very much. And using these techniques, we've been able to produce a model system of how the water column operates. And what is important here is that it's a system driven by the iron cycle. You can see here the acronyms of different microbes involved. Depending on the size of the acronym is the abundance of this microbe in the system. And what is important is to underline that is a system that works in the aerobic and the anaerobic part, and centered in the iron cycle. Also, a river has sediments. So a few years ago, we started to be interested into study the diversity in the anoxic sediments of the river. And as you can see very easily, there is many more microbial acronyms. This means that there is more diversity in the anoxic sediments of the river. And this is probably related with what happens in a system like a sediment, that the matrix is semi-solid. And we will see that in a solid matrix, this is even more interesting. We also pay attention to the geological part of the system. In the system, there is a lot of iron mineral deposits that are preserved in sedimentary rocks. Here you can see some of the minerals that are trapped in this kind of structures. In the upper part, you see the famous jarosite. Jarosite became famous because it was detected on Mars. In the middle part, you can see spermanite and goethite. And the lower part, you see hematite, the most oxidized iron that you can find. But when we try to date the iron deposits, the oldest iron deposits, that we know that they are the oldest because they're the ones that they are in the upper part and the upper part of the river, it turns out that they were over 8 million or more years old. And for many years, and you can find it today even in the literature, in the references, people thought that the origin of the Tinto River was the very heavy mining activities that have been operating in the area for more than 5,000 years. But you can see that if you compare the 5,000 years with 8 million years, there is a lot of gap between the two dates. So this means that we have to try to find another origin for the river that is not related with the mining activity. And one possibility, and inspired by the Darwin's suggestions, I mean, we work with the hypothesis of the existence of an underground bioreactor, which, knowing that there is a high concentration of metal sulfides and there is water on the system, I mean, microbes that are able to take advantage of this situation, produce reactions that they drain to the river, and they produce this acidic river with high concentration of fluoric iron. And if you want to really know about that, you need to drill. Most of the work in the subsurface has been done with groundwater obtained from artesian wells. I mean, it gives you very interesting information on the diversity, but it doesn't tell you any information about the interaction between the microbes and with the minerals in which they grow. Also, some subterranean mines have been used for the same purpose. But again, my personal view is that constructing the tunnel for the mine, I mean, this is changing the microbial ecology of the system, and whatever you can obtain from the walls of the mine, I think it's not meaningful from my point of view. So that's the reason why we prefer to use devoted perforations just to control all the situation and to trust that all information that you get, it's the native, it's from whatever exists in the subsurface. So a few years ago, we applied for a ERC advanced project, and we were granted. The name of the project was Iberian Pilot Bell Subsurface Light Detection. And what I will do today is just report what we learned with this in this drilling project. So here you can see a view of the drilling place, the part of the area of the Rio Tinto that is called Peña de Hierro, Iron Mountain. And you can see also something that is very important. The most important instrument for geomicrobiology, deep subsurface geomicrobiology, is the drilling rig. And also you have here a core in which it's marked the presence of massive pilot that is very important in the system. We use plastic liners in the drilling to avoid an excess of contact of the drilling fluid that is needed for drilling. And we introduce in the drilling fluid sodium bromide as a marker to know which samples have been in contact with the drilling fluid and probably contaminated. After the cores are recovered, they are cut in pieces that are easy to manage. They're put in plastic bags. You inject nitrogen to remove oxygen. Remember that under our feet in the subsurface, there is no oxygen. It's an anoxic environment. And all the work has been done in anaerobic chambers just to avoid the contact with the oxygen. And here you can see how you obtain the samples from inside of the cores. Once an interesting place is located, with an hydraulic press, the core is cut, and then with a drill at lower speed and controlling the temperature, you get a sample from inside of the core. Here you have a profile of the minerals through all the drill. I will not get into the details because I'm a microbiologist, not a geologist. But at the end of the drilling, what you can do is to get a profile of the temperature, of the diameter of the drill, and also the radiation, the gamma radiation. The gamma radiation probably will be interesting in something that we will discuss later on. And all the samples that has been deeper analyzed biologically, geologically, and geochemically. I mean, we do a thorough XRD analysis in order to know with which minerals the microbes are in contact with. From previous drilling in the area, we learned that it is very important to decide which cores are interesting to analyze in detail. And the best way to do that is doing an ion chromatography analysis just right off the drilling. And doing that, I mean, what you can see here in this case is a display of nitrate, which is very interesting. It's almost constant and present in all the borehole. It's not the same with nitrate, phosphate, and sulfate that they have a stochastic distribution along the borehole. But yet, even today, the geologists still don't understand why the presence of this constant concentration of nitrate along the borehole. The presence of nitrite, and also ammonia that is not displayed here in this figure, it claims that probably the nitrogen cycle is very important in this drilling. Also, phosphate is a good news for microbes because they need it for their nucleic acids, etc. And sulfate is a way to know that the metal sulfides, mainly pyrite, it's being leached. So in other words, there is a process that is just producing sulfate from this pyrite. Also, with the same technique, what you can get is information from the short chain organic acids, mainly acetate and formate. And this is a good clue that there is biological activity underground. Also, we did gas chromatography analysis, and we detect along the borehole hydrogen, CO2, and methane. The first two, hydrogen and CO2, are good news for microbes. One, because it's a good electron donor. The other one is because it can be used as an electron acceptor, but also is a good source of carbon for autotrophs. The biochemistry allows to detect the stochastic distribution of proteins and sugar along the borehole, and also doing next generation sequencing. What you can detect is the diversity, just checking out the sequence of the 16S ribosomal RNA gene. I don't pretend that you read anything, it's just to let you know that we get this kind of information working with Illumina. And here you have a close-up of some interesting data on the sequencing, in which you can see the detection of microbes that they can use hydrogen to grow. We know that there is hydrogen in the system, and also microbes that are able to use nitrate for anaerobic respiration, oxidizing iron. Being in the Center of Astrobiology, we've been able to use the immunological chip that they produce in order someday to fly to Mars and detect signs of life there. But while we are waiting for that, we can use it for other purposes on Earth. And here you can see in a sample at minus 538 meters that we detect the presence of sulfobacillus and Shevanella, as you will see by other techniques have been also detected. And a signal of Pyrococcus furiosus that was not expected because the last volcanic activity in the area was 340 years ago. But yet, remember that this is an immunological detection, so this means that you have the structure of the antibody that is being recognized by the antibody, not necessarily this type of microbe. But as I mentioned before, one of the techniques or the methodologies that I like the most is the fluorescence in situ hybridization. Here you can see the signal, the hybridization sign at minus 415 meters below surface. In the upper part, the hybridization signal with beta proteobacterial probe. In the right, with a gamma proteobacterial probe, and also on the bottom, a signal for Acinetobacillus, which is very is a type of a microbe that it's very in general identified in acidic environments of this sort. And here you have a display of the different hybridization signals that we detect by CARFIS. CARFIS is a modification of the FIS methodology that amplifies the signal. So this allows to better detect the hybridization signals and also eliminate the artifacts that they can be produced, because most of the minerals that you have in the subsurface, they are autofluorescent. So they are also giving some signals that have to be detected and eliminated. And one of the interesting things from this type of data compared with different variables that we have in the system, it's just that we don't see a gradient that usually is described in many other drilling systems. Probably this is because the characteristics of the Iberian Pyrite Belt or something that still we are working on it. And now I will ask you to pay attention to the colors of the different probes that we use to detect different microbes. Here in red, for the size of the bar, it's 20 micrometers. Well, you will see in red the hybridization signal of for acidoborax, specific for acidoborax, which is a microbe that is able to oxidize iron. For the same sample, now you see the signal for acidophilium, which is an iron reducer, and for the same sample, the signal for sulfobacillus in blue, which is a sulfur oxidizer, and in green for sulfur reduction. And if you use a confocal microscope, now you can see the merging of all these different signals, and you see something that was expected, but has to be proven, that the iron and the sulfur cycles are very close, or they are collaborating in the subsurface of the Iberian Paya Belt, which I think is an interesting observation. Also, the same technique, we use the appropriate concatenable a stain that is able to detect exopolysaccharides. You can see that most of these microbes, they are not alone in contact with the minerals, but they are embedded in biofilms, in exopolysaccharides, making biofilms. And here you can see two different detections at different depths, and you can see the red and signals for for bacteria, and in blue for archaea. And you can see that they are hybridized together. So this means that again they must have something in common, probably interchanging metabolic products. And nearby you can see the signal for polysaccharides, and the last one is the merge signaling, which again you can see the microbes that they are embedded in this exopolysaccharide matrix, in other words, in biofilms. It's very interesting because in the literature you can find that many authors claim that oligotrophic subsurface environments are so poor in energy that microbes cannot afford to expend this type of energy for producing biofilms. So this kind of work shows that that's not true. Really what happens is that if microbes need biofilms, that probably they are good to facilitate the control of the metabolic products or the interconnection between the micro niches in the hard rock system. I mean, they just spend the energy that is needed, and they go and they do it. Also, we use enrichment cultures. Enrichment cultures is a fast way to know what kind of metabolic activities might be on the system, and we check for pyrite, iron, and sulfur oxidizers, also for iron reducers and sulfate and sulfur reducers. So I mentioned that before, the iron and the sulfur cycle. We detect methane, so we look for methanogens. If methane is produced, it will be wise to use it for biological purposes. So we look for methanotrophs. Also, because we detect a lot of nitrate, we will look for the nitrifying bacteria, and also because we detect a lot of acetic acid, we look for acetogens, which, by the way, share with the methanogens the same kind of metabolic elements like hydrogen and CO2. If you have enrichment cultures, you can isolate microbes, and that's the list of the microbes that so far have been isolated in the deep subsurface from different enrichment cultures at different depths. And the names that you have here, the species names that you have here, are the close relatives that the sequence of the genome, the 16S, the sequence of the 16S ribosomal gene gives you as information. And now, putting all this information together, here you have a table with a display of the 26 more abundant microbes at that general level that we detect at 20 different depth intervals. And in the lower part in blue, but you can see the different techniques that has been used in the different depth for detecting this type of microbes. We, in our experience, we've been critical with the many of the drilling projects that have been published because they use only one technique, generally sequencing, to describe the diversity. And we know that sequencing has bias like any other technology. So we thought that maybe a better approach to describe the diversity of the system was not to rely on one methodology, but to use all of them and put the following straight. I mean, we select the most important genera working in the subsurface of the Iberian Pyrite Belt if they follow this criteria. They should be at least identified by two independent. Independent is very important because they should be based on many different principles, and not only by two independent methodologies, but also they should appear at 5 different depths of the 20 that we analyze. And doing that, here you have the 16 ones that they accomplish this requirement. And on red, you can see the ones that they have been isolated, so we can always sequence their genome and know the type of genes offer that they have. And doing that, you can see here the comparison of the results obtained at 8 different depths for different metabolic pathways related to the nitrogen, the carbon, the iron, the hydrogen, and the sulfur cycle. And you can see that even though they are not all the same, you can see differences in the number of genes that you can detect. I mean, most of these cycles, I mean, you have the means for these cycles to be operative. And this gives us this model system that we have today, I mean, these days, in which you can see the couple carbon, hydrogen, nitrogen, sulfur, and iron biogeochemical cycles in the subsurface of the IPB. In blue, you can see the nitrogen cycle. In red, the iron cycle. In green, the sulfur cycle. In the upper part, you see the carbon and the hydrogen. And in the middle, you can see electron donors, electron acceptors that has been detected in the subsurface of the Iberian Pyrite Belt. In red, you have the microbes that they have been isolated, so we've been able to sequence the genome and annotate the genes that are responsible of these specific reactions. And remember that what we wanted to understand was the origin of Rio Tinto. So we need to understand how the system operates in order to produce ferric iron in acidic solution. So the first experiments we did is just add water to some of these core samples and follow with time the production of iron. And you can see with different efficiencies at these 3 depths, we can see oxidation of iron. But when we check the genomes of the microbes that we isolated, we couldn't see any that had the genes that has been described for iron oxidation. So we have to conclude that the probability that this oxidation was driven metabolically, I mean, it didn't sustain. So we come out with the parallel idea that maybe what you have is an indirect way to oxidize iron. As we saw, there is a lot of nitrate in the system. We know that there is a lot of nitrate reducing activity. So what we check is the possibility that the product of the nitrate reducing reduction, which is nitrite, which is a strong oxidant, can oxidize iron. So we tested for 8 of the isolates that we already have in our lab. And what you can see with different efficiencies, I mean, the ones that are on display, they can oxidize iron through nitrate reduction with different efficiencies. I mean, also what we try is to see if this ferric iron can solubilize, oxidize pyrite. So some preliminary experiments that we are following, I mean, they are not finished, show that with a specific microbe, acidoborax, with the presence of iron, as acetate and nitrate, I mean, this is acetate is the electron donor for acidoborax, and nitrate is the electron acceptor for anaerobic respiration. We can see that in control conditions, we can detect release of iron from Biden. So that's the reason why we call it that preliminary, because it needs much more work. But we think that that's the way that the system gets the ferric iron that finally drains the drain in the river. And this is the lessons we learn with this work. As you can see, we detected a high level of functional diversity in the subsurface of the IPB up to 620 meters. That's the limit of our drilling. For the first time, we've been able to understand how the most important biogeochemical cycles operate in a coupled mode in the subsurface. The nitrogen cycle seems to be most important than we thought previously, that different nitrogen-reducing bacteria have the ability to oxidize iron in strict anaerobic conditions. And all these results together show that the hypothesis that we had, that probably there is an underground bioreactor that is able to originate what you can detect in the basin of the river. I mean, it is true. Also, one question that we ask is, how similar is the Iberian Pyrite Belt subsurface with other systems? We know that the Iberian Pyrite Belt is very peculiar in terms of mineralogy. So what we compare is the diversity I just showed you with the results that has been reported for 18 different continental deep subsurface drilling operations. And you can see at the phyla level that there is differences, but the differences are not that important. I mean, what you can see that all these phyla, members of this phyla, are present in these different drilling conditions. This is, for us, strongly suggests that probably the dark biosphere is more homogeneous at the diversity level, and hence as the operation level, than what we thought before. So this might be, they are independent on the location, the mineralogy that you are drilling through, and obviously this is a field that it will need much more information in order to prove these interesting hypotheses. And one obvious question is, well, what is the interest of this type of work? And well, one side is the economical one. This is used these days for biohydrometallurgy. It's a way you can obtain many metals around the world. I mean, probably that's the mining, the future of mining for our society. But I will not talk about this because this was not my interest. My interest is astrobiology. So I would like to know this kind of information, how important is, for instance, in Mars habitability. And well, we know that different missions reported different interesting things that are related with the kind of data that I just showed you. For instance, the mere opportunity in working in Meridiani Plano was the first one to show the presence of jarosite. Jarosite is a potassium sulfate that in Rio Tinto is produced biologically. So it's a huge biological activity. In Mars, we don't know. We know that there is jarosite, and now there is the open question if this has been done biologically or there is other ways to produce jarosite independently of the presence of biology. Also, the curiosity, Robert was able to, heating different samples, produce compounds, molecules that we've been detecting in the subsurface of the Iberian Pyrite Belt, like CO2, like hydrogen, and also different states of oxidation of sulfur, meaning that there is the possibility of a sulfur cycle. the perseverance, we are still waiting for the decision of how we recover the samples that we produce, and in order to do a thorough analysis on Earth and see if there is signs of life and try to answer this question. But if we check the life requirements, which we know that they are very basic, you need a source of energy, the basic elements used for life systems that we know, and also a solvent, an adequate solvent to facilitate mass transfer reactions. Of all these three, we know that in the subsurface of Mars, there is all the components that we also detect in the subsurface of the Iberian Pyrite Belt. By the way, I forget to mention that today is considered a very good mineralogical and geochemical analog of Mars. I mean, you can see that all these molecules have been detected on Mars. They are related with whatever happens in the subsurface. So this means that this type of metabolism, it's possible using this data on Mars. But what we need is the solvent. For many years, I mean, we knew that in the surface of Mars there was no water. Mars lost its water, but in a report 7 years ago, the Mars Express was able, using his Mars's radar, detect the presence of liquid water in the subsurface at 15 kilometers depth. Later on, they just found this time the same kind of water at different depths on the subsurface of Mars. And recently, just using the data of INSIGHT, a mission that was devised to get geophysical data from Mars, I mean, some authors were able to use the information for this mission to show the possibility of important amounts of underground water on Mars. So this means that the most critical element that has been looking for in many different missions, I mean, exist in the subsurface. So, we have to conclude that knowing the surface of Mars today, strong UV radiation, oxygen conditions, and low temperatures, and absence of water, we don't think that is the ideal location to develop life. But knowing the possibility and the properties that we already know from the subsurface, and the most important one is the presence of liquid water, is that there is many more possibilities to detect life on the subsurface if ever exist. And we are dreaming on have a devoted drilling mission if we want to really know if there is life on Mars. And what happens in the universe? Well, a rough calculations on the number of galaxies and the possibility of planets that every star on the galaxy has, we can arrive to a very important number of exoplanets that we know that they must exist in the universe, but still we are in around 5,000 and a half detection mode. And this allowed me to go to something that I want to share with you. My worry about the very conservative habitability concepts that we are using today in astrobiology, in astrophysics, of course, but in astrobiology. And this is what we call the habitability zone that was generated by Casting and collaborators in a seminal paper in 1993. But now, today, I mean, and the limitation of this concept is that the calculations were done for planets that they can hold liquid water in the surface. I mean, just Mars, without going any farther, doesn't have any water in the surface. They probably had it a long time ago, but today it doesn't have it. But if you know, we know that in the only model system of life that we have on Earth, there is life in the subsurface, we should start to think that maybe what we need is an habitability zone concept which incorporates the subsurface. And Mark Mahon, in a paper a few years ago, 7 years ago, described what is called subsurface habitability zone, or in other words, the SSHZ, which I think in the next figures you will see he used the same analogies and the same calculations that Casting used for his paper, but in this case, he is looking for life in the subsurface, not in the surface. And here you can see the possibility of the existence of life at different depths between two ranges of temperature, 0 degrees and 122 degrees. And obviously, this will be very dependent on the distance of the exoplanet from the star. He also did the calculations for different masses because this is dependent on the mass of the exoplanet. And you can see that depending on the masses, there is different types of lines. And this comes to my conclusion that once life on the subsurface on a rocky planet has been demonstrated, our extremely limited concept of habitability zone must be broadened. We suggest incorporating in the Mark Mahon subsurface habitability zone in future exploration missions, evaluating the possibility of the existence of water in the subsurface, the only limiting requirement for life. And with that, I mean, the growth, amplify the possibility to detect life in the exoplanets and the rocky planets, of course, and in the universe. I mean, this claim has been described in a paper that we just published in Frontiers last year. And with that, I think I arrived to the end of my talk. And again, I hope you enjoy it. And I hope that this inspires you to put in your thinking that subsurface, the dark biosphere, the subsurface biosphere is very important if we want to understand how life operates in the universe. Thank you.
Yes, now there are two possibilities for all to ask questions. Either you raise your hand or you ask a question via the chat, which I can also see. The floor is yours. Nick, how do we effectively search for life in the subsurface without contaminating it with terrestrial life? A very important question.
Well, the technology to do that on Earth, it's available. I mean, the only thing we have to learn how to do it going around searching for life. The methodologies exist. What the most important thing is the way you operate and also to have contamination control. I mean, something that allows you to know if your sample has been in contact with something that could contaminate the sample.
Another question? Well, I do not really see anybody. Just let me make my...
Hi.
Hi, yes.
Hi, Wolf.
Josep, yes.
Okay, first of all, congratulate Ricardo for all this amazing work. Really exciting to see all these microbes in the different slides that you made. I wonder, in view of this evidence for, deep surface liquid water in Mars, if you agree with that observation. Do you think Mars will be the first planetary body to discover these microbes?
Answering your question, I was very lucky to be in a course in Italy, and in the audience there were the authors of the papers. So we had a lot of time to discuss and all the problems that they had to solve, because you know many of these observations were done a long time ago and it were not easy to publish. But I think today, at least from what I understand, I'm a microbiologist, not a geologist. I mean, for me, we're convincing. And I think the recent data on InSight is a good backup. You know, I mean, there is not only one type of observation, there is another based on a very different methodology that shows that that's possible. I mean, and going back to your second question, I'm a strong believer that we will find life on the subsurface of Earth. Hello.
Are there any other questions? If not, I would just like for 1 or 2 minutes make an advertisement, namely for our next big conference, namely the Beacon Conference in Iceland in July. I hope that you can see my screen now.
Yes.
And yes, Beacon has been last time in La Palma in the Canary Islands. This time we will go to another volcanic island, Iceland. We will have 5 days of scientific program and an excursion or 4 different excursions to geologically interesting sites for you to choose. We also have a trip after the conference where we'll explore a little bit of southern Iceland. There will be also an early career scientists meeting, and for members of the European Astrobiology institutions, there will also be bursaries. I think we not only have very interesting program, we also have some excursions to places like geysers, waterfalls, interesting geological formations, active volcanoes, and last but not least, dinner in the Carousel restaurant Perlan with very nice views over Reykjavik. I hope that I can see you all in Reykjavik in Iceland, and I hope also that you will join our next seminar, which will be in exactly 2 weeks. Up to then, have a nice time and see you at the next